Now we're going to talk about DNA replication. And here, this schematic show the basic of DNA replication. Here on the top, you can see that the DNA is together. You have one and two strands, one, and this is the other strand. And these strands are held together by hydrogen bonds. And remember that I told you the hydrogen bonds were a type of bonds that they were very easy to break. So an enzyme can easily pull apart the DNA and break into two pieces, break it into two strands. And we can not only do that using enzymes, also temperature, just increasing the temperature of DNA, the hydrogen bonds are going to break, and this will allow the DNA to be replicated. In the third schematic, you can see that the old strands are here and a new strand appear. And how these appear? They are just by DNA complementarity, you can copy and you can make an exact copy of the other strand. And the same thing happened here. You have an old copy and a new copy that is attached, is added just by DNA complementarity. So to summarize, the two strands are complementary, each act as a template for building a new strand during replication. And in DNA replication, the parent molecules unwind, they separate one from each other, and the two new daughter cells, that are in this case the light blue, are built based on base pair rules. Unless now that we know how the DNA replication takes place, let me walk you through some of the experiments that allow us to find that this was the correct model. In the beginning, there were three models. There was the conservative model, and there was the dispersive model, and also the one that it was right, it was the semi-conservative that I will discuss in the next slide. But so let's talk first about the conservative model. And in the conservative model, I want that you remember that this is not right. And from the dispersive model, I want to remind you that this is not right. But let me explain it why. In a conservative model, imagine that the DNA, this molecule, when you wanted to copy it, when you want to replicate it, you are just making a photocopy. So you are going to have an original, and in the next generation, you're going to have an original, and you're going to have a copy. And here you're going to have a copy and another copy and another copy. Yeah, so you're only keeping an original, and there is no mixing between the two strands. So this is the conservative model. So the conservative model, the two parent strands rejoin all the time. So here, these are going to rejoin all the time. And this is a new strand. Yeah, and this is a new strand. Now let's talk about the dispersive model. In the dispersive model, if you see here, instead of being being a photocopy, is like when you have a text and you just copy and paste pieces of the text. Yeah. So imagine this is the original in dark blue, and you in the first generation you have pieces, one piece that you copied. Here you see this piece you copied in dark blue. Here you find another piece, and the same thing happened here. You already copied this one, and you copied this section. However, in the light blue, you find other sections that are new. So, and in the second generation, something like that happened. So you have old versions, and you have also here an old version, and that is the light blue here. This one will be the light blue. And in the next generation, you're going to keep the old one, yeah, in dark blue. Here, the old one in dark blue. And you're going to have a new one here and a new one here. So you only keep one strand. So Watson and Crick, semi-conservative model of replication, predict that when a double helix replicates, each daughter molecule will have one parental and one newly made strand. And this one, as I said to you, this one was the correct one. However, at this point, it was just a hypothesis. To test this hypothesis, Meselson and Stahl had to make a very sophisticated, and some describe one of the most beautiful experiments in biology for some of them. And how they did it, they use an atom that we know that is called nitrogen, and nitrogen generally has 7 neutrons and 7 electrons and 7 protons. And sometimes you can get isotopes. An isotope of a nitrogen is, instead of being a nitrogen 14 that has a mass of 14, here you have a nitrogen of 15. Why? Because you have an extra neutron. So it's a little bit heavier. Yeah, instead of having 7 neutrons, you will have a neutron. So instead of being nitrogen 14, it will be nitrogen 15 because have an extra neutron. And if you remember, the nucleotides, the adenine, the thymine, the guanine, and the cytosine, they have nitrogen. You have a nitrogen here, you have a nitrogen in there, and there are so many nitrogens. So what it means that you can make molecules of DNA that are heavier than the ones that have this one that have the isotopes will be heavier than the one that have the normal nitrogen. And this will be key to differentiate between the three models. And you can get light molecules of DNA here. You can get light molecules. So for example, here, and I'm going to show you that identifying how big is the when you separate using mixing them using a machine called centrifuge, you can separate them, and if you have a lot of DNA or different amounts of DNA but on the top, it can be a good indication that the DNA has a lot of the nitrogen 14, and if you have an intermediate, it could be a combination between both 14 and 15. And later I can show you that if you have the heavy one is the one that is going to be in the bottom. Is going to be in the bottom, so you can differentiate the three models using isotopes, an isotope of nitrogen and a normal nitrogen, and it will give you different bands doing an experiment. Now I'm going to show you, I want you to watch the following video that explains the Meselson experiment, and later on I will explain in a little bit more detail the analysis of this experiment.
The Meselson and Stahl experiment provides evidence for semiconservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Meselson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So now let me walk you through the experiments. And this experiment has 4 steps. And just to remind you, Matthew Meselson and Franklin Stahl did this experiment and showed that the semi-conservative model was the one that it was correct. And they did it by using a heavy isotope of nitrogen. And they found that the old strands will have the old isotope, while the new strands will have the light isotope. They start the experiment by culturing in the first section of the experiment, this section, they grow the bacteria in a medium that has the heavy isotope. So they wait for several generations and they grow the bacteria there. So that every time that the bacteria need to make new bases, it start absorbing the heavy isotope. And then that way they make sure that all the DNA in these that have these bacteria, they had the DNA with a heavy isotope. And then to go to the second step, they move it to the lighter isotope. They move it to food that have the lighter isotope. And remember, so this, as the bacteria start eating that food, it start absorbing the new isotope. And every time that the bacteria divide, is going to start adding the lighter isotope. So they, as they know that the bacteria divide every 20 minutes, so you can have different cycles of replication. So if you want to see what happened in the first cycle, you have to stop the experiment at 20 minutes. If you want to see what happened in the second cycle of replication, 2 rounds of replication, you stop it at 40 minutes and you continue doing this experiment like that. And why this is helpful? Because later they can centrifuge, yeah, they can mix, and it will allow them to see the density of the DNA. And if they have only one band, if they only have one isotope, they will find a band. And if they have a combination between the heavy isotope and the lighter isotope, you can have 2 bands. Yeah, and in that way they can figure out which one of the models was the correct one. But let me walk you more through that. So here, all the bacteria have the heavy isotope. So as a result, here you can see that all the bacteria have the heavy isotope. When they check the first generation, as this is the semi-conservative model, they are supposed to find one that it was heavy, that it was the red one, yeah, one that it was heavy, and the other one that it was light. So that is the blue one. So here, light and blue one. So you are going to have one that is 14 and one that is 15. So you are going to have an intermediate. So in this case, this matches the semi-conservative. And I will show you in few slides that it doesn't match the dispersive, and it doesn't match it's not consistent with the result that you expect in the other 2 models. And then if you check the second generation, in this next generation, you're going to have the old one that is red, and you're going to have a new one that is blue, and you're going to have 2 that are new one that are blue, and the same thing will happen here. So result, you're going to have this that is a combination. Yeah, and this is what exactly they found. They found one with an intermediate, but you're going to start finding also blue one that are lighter. So half of them will be here. You are going to have half of them, 50% of them will be light, and here 50% will be intermediate. Well, in the beginning you had 100% intermediate, Sorry, this is not a pretty 100%, but this one is 100% intermediate. This one is also 100%. And in the third generation, if you look here, you can tell me what is happening. You can think what is happening. What will be the proportion? So you can stop the video if you want. But what is happening here is like, look, you have a lot of new molecules of DNA with the lighter isotope. And as you don't have any more heavy isotope, you start having very little that is intermediate. You have only two of the molecules that have intermediate, while one, two, three, four, five, six of them. So you have three more times more the light isotope, and you have a smaller band of intermediate. But if you see, you lost the heavy one. Yeah, there's no heavy one anymore in the first generation. There is not more heavy one in the second or in the third generation. And in this way, Meselson and Stahl, they found that the replication was semi-conservative. So they, in the first generation, they found that all of them were heavy isotope, while in the second one, it was a combination of both. And in the third one, you have the lighter one start appearing, and you have 50% of them that it was intermediate, and later the amount of lighter one will continue increasing and increasing, and if you continue to fifth, sixth, seven, the the one that is lighter will start increasing, so this one will increase while the intermediate will reduce. Good. Now I want to show you what happened, and I want that you think, what do you think that happened if the model was conservative? So, and I'm going to show you an schematic that explains what happened if the conservative model was right. And this could be a great question for the test. What do you think what were Meselson and Stahl experiment expecting if the conservative model was right? And I remind you, in this conservative model, it looked like a photocopy. So you make this one, it was the heavy isotope. This one was the heavy isotope. You make a photocopy. You make a photocopy, and all of them are the originals, while the other ones are just copies. So if you see here, you are always going to keep this heavy one, and it's a very thin band. So if the conservative model was right, you're always going to keep this one, a thin band, a very slim band of the heavy isotope. Well, as the generation start increasing, you start increasing the amount of new light bands. And is what is interesting here is that you never found an intermediate. Yeah, this N14 and 15, it never take place in the conservative model. Okay, so if I ask you that question, you know that the answer will be that in the conservative model, if the conservative model was right, you're never going to find a combination between the N14 and F15 band. And now let's think what happened if the dispersive model was right. This could be also another question for the test. And what do you think that what will be the result that you're expecting? And here I just remind you, in the dispersive model, you will start again with this heavy isotope, but later you start making copies. You start mixing both the heavy one as well as the lighter one. In one strand, you will have both. So as you continue generation through generation, you are just going to find a mix between all of them, pieces and pieces of the N14 and F15. So you will find in the first generation, and in all the cases, you will find in the conservative, semi-conservative, and dispersive that you start in all the cases in the same way. You have a lot of all of them are N15, the heavy one, but in this case, you start having a mix, yeah, between the old one and the new one. Yeah, maybe in the future this starts slightly moving more in this direction, getting more into the N14, you start having more blue pieces, but all of them are mixed. So this is not what we found. This is not what we found in the in Meselson and Stat. Found very different results. So this is why we know that this is not correct. To summarize, we found the predictions that of the three models show that the conservative and the dispersive model are not correct. Only the semi-conservative model is correct. And we analyze what were the expected results in the first and in the second and in the third replication. And you could go back in the video and try to look, and then maybe you can stop the video and try to draw them, try to understand what is happening. But remember that just the first replication produce a hybrid of DNA. So you will find this is a hybrid. One strand will have the old one and the other one will have the new replicated. So this eliminate just right away that the possibility of a conservative model, because here, as we talked in the previous slide, you're just going to keep the heavy one and you're going to make a new one, and this is not what happened. So this was the way to remove it. And the second replication produced both light and hybrid. So here you've the light and the hybrid. Here are the light and the hybrid, one old one and one new one. And these eliminate the possibility of a dispersive model, because here you're just going to although you start in the beginning, the dispersive start looking a little bit similar to the semi-conservative. In the light one, in the dispersive model, you're just going to keep producing more and more molecules that look like hybrid, and this is not what you see in the experiments. Finish. So in this way, Meselson and Stahl test the hypothesis of Watson and Crick and show that only the semi-conservative model of replication was correct.